there's my disclosure slide. I'd like to thank the organizer for inviting me to present today. Nature Technology is a technology company that on the left shows some of what we do. We have vector technology and we have production technology, and we offer it as services and or licenses to industrial and academic clients. On the left side shows some of the stuff that we can do. In the process of doing these services, in the process of doing all those kind of services, we've seen thousands of vectors. And the one I'm going to talk about today is general vector design criteria that can be used to design better vectors. And I'm going to focus quite a bit on indirect clinical vectors because this is more of a virus meeting and a transposon meeting. We do get a lot of transgene design. I'll give a little bit of an overview on transgene design. In the second half, I'll talk about designing promoters. But most of our technology is around the backbone. And so I'm going to really focus on the bacterial region. And so this schematic on the right is just your typical non-viral vector. And that bacterial region is really underrepresented in terms of research interest. The eukaryotic region people are very familiar with, the promoter, the transgene, the poly-A, and then the viral or transposon termini adapters that are needed for those kind of vector production. So I'm going to start off talking very briefly about production. One of the technologies we have is a fed batch fermentation process, and this is called the HyperGROT process, and it is a reduced growth rate at low temperature followed by a temperature upshift to 37 to 42, either fast or slow depending on the virus or depending on the vector that we're manufacturing. And this delivers very high yield production, and a lot of you who are going into the clinic may have encountered it because we have it licensed to a number of CMOs. Now, downstream purification, this is a schematic. Most of the purification you're going to see is the top line, which is alkaline lysis. And the reason I'm showing this is that often when people do their preclinical work, they're using a single downstream purification step. For example, the single chromatography and anion exchange chromatography, like a QIAGEN kit. And typically then you go into GMP manufacturing, you end up doing at least 2 purification steps. And you're going to meet, this is work we did with VGXI, showing the kind of specifications in terms of purity for impurities like genomic DNA and endotoxin that you meet when you do the 2 purification steps. This is significant just from a development of your product point of view, because the impurities you'll have in a QIAGEN prep, which a lot of people are using for their development work, can lead to things like activation of dendritic cells, can really interfere with formulation. So you've heard talks on putting together plasmid and polymers together. That can be highly impacted by impurities in the DNA, which can really affect your performance. And then they actually, even without formulation, you can cause damage to muscles or to the lung through these contaminants in there. So you hear this a lot in industry, your process is your product. Think about the purity when you're going forward with your using results from animals, because if you don't have pure enough DNA, you may end up having a result that's not indicative of what a hyper-pure product would give you. I'll come back to that a little bit later when I talk about minicircles. Okay, very briefly, I'm going to talk about codon optimization. So basically, people come to us with vectors, and often they've done the top part where they've defined the transgene sequence. You see, a lot of times people will swap out the signal peptide for a more designer signal peptide to get higher yield expression, because secretion is a limiting factor. So with a lot of things that you'll see things like the TPA signal peptide. You also see stabilizing mutations in the gene therapy field. You see the factor 9 Padua mutation, which increases the activity. It drops the level of transgene expression you need to be able to get a therapeutic effect. The next 2 boxes, by incorporating design criteria and removing design criteria, are often handled really just with an online resource like the GeneArt program. So that's program actually does both the additions and the subtractions. So you can use GeneArt to design a codon optimized gene, which generally gives you higher expression than the native gene, which is why people are using it. And you see on the bottom right, the pricing is, and the time delivery has gotten so good that that's just generally how people do it now. The other thing is just as an aside, a lot of times people, like a company will in-license vectors from a university, and then the parts and pieces might be subject to material transfer agreements or something like that. You can basically get a very clean regulatory report by just making things on their own. And that 18 kb is not a typo. We've just recently, people who don't want to have to figure out where all their parts and pieces came from, simply whole genome sequence their vectors and just had it us make all their vectors de novo. A cautionary note about codon optimization, I don't see a lot of people thinking of this, so I'm really mentioning this and highlighting this, is that this idea that your translation starts at the Kozak ATG and then goes to the stop codon, and that's all you make from that messenger RNA is not accurate. There's cryptic ORFs that are expressed from alternate ATGs and alternate start codons, and there's a lot of literature that shows there's enough of those ORFs made to get a T-cell response. So when you make a new codon optimized gene, you have different ORFs in there, and you have to make some careful with that because those could give you T-cell responses even though you think you're making a native protein. And another thing about codon optimization is you've got to be really careful with your single peptides and GPI anchors, that some of the rare codons that are present in sequences can be there for a reason, to slow down translation and allow things to fold. So often you see folding doesn't work properly after codon optimizations. You've got to be very careful in those regions. Those are the only ones I know of that the rare codons seem to be an issue. All right, so there's two types of vectors that we deal with. I'm going to talk a lot about the indirect gene transfer vectors today, but there's the direct and the indirect. So the indirect are where the plasmid is used to actually make the product, but the plasmid isn't actually part of the product. And so this would be viral vectors like AV or lentiviral vectors and transposon vectors. And then the direct gene transfer vectors are the ones like you heard in the first talk, where the actual vector is the product, where you're trying to do a gene therapy, for example, or DNA vaccination. So this is basically what you see in the maps when you look at the literature of these indirect gene transfer vectors. And on the left you have AV. You see there's 3 vectors. On the right is on the center is the lentiviral vector. That's a 3rd generation where you have 4 plasmids. And then on transposons you have 2 plasmids. And in the cartoons, it's always just showing the eukaryotic region. And if you look at the center one, you'll see the whole bacterial region is just drawn as a black line. And then on the bottom right, I've blown up from one of the catalogs what this looks like. And you see about half the vector when you look at the LTR where the right and left LTRs are. About half the vector is this bacterial region, which is just supposed to be selection and replication. So where does this come from? I've seen thousands of these things. Virtually all these bacterial regions come from the 80s. And I don't think I've seen one that hasn't, doesn't have origins back there. You see the blue script plasmids, you see the puck plasmids, you see all sorts of vectors. Now, just because something's from the 80s and it's old, 30 years old, does not mean it's bad. You can be old and still have some value. But so this minicircles were developed as a way, for a number of reasons, but one thing that they serve is they're a research tool that allows you to get at, is that old bacterial region a problem? And so I'm not going to suggest anyone go into the clinic with a minicircle. They're very difficult to make and hard to get GMP, but they do allow you to precisely remove the backbone and say, what effect does it have? And so this is work that really started off from Mark Kay's lab at Stanford. He's done a tremendous amount of work on this, which is really illustrated, especially in tissues like the liver, which are prone to silencing, that a backbone over a kilobase will cause profound silencing of expression. So that basically the vector will shut off. And they've shown that it doesn't matter whether it is bacterial in origin or just spacer DNA. It's basically, if you take the Bollier, which is where the transcription stops of the vector, and then go to the promoter, if you have more than a kilobase between those, you're going to get silencing. So that's in vivo, but what about in indirect vectors? So this is results from 3 groups that have shown that minicircles with a transposon vector, which is now you're doing ex vivo nucleofection of primary cells. They've shown much better effect with much better sleeping beauty transposition and reduced toxicity with vectors that don't have a backbone. So in this case, they're getting 6-fold higher CAR-T production using a minicircle backbone, probably due to the absence of transgene silencing and transgene toxicity. And this is more of a practical point of view. People have recently shown that AAV vectors used, it turns out that again, as I talked about with translation, AAV packaging isn't quite as simple as it goes from one ITR to the other ITR. You do occasionally go beyond an ITR. So recently people have shown that you're actually packaging up the bacterial antibiotic resistance marker and transferring it to patients using a standard backbone vector. So there's a number of issues with the backbone that's been gotten at by looking at minicircles. So our platform is a nanoplasmid, and as I mentioned, minicircles are hard to make because you have to do a lot of in vivo recombination. Nanoplasmid is just a plasmid that has a short backbone that allows you to eliminate that toxicity, the gene silencing that the larger backbones have, but has high yield manufacturing. So it has 3 components. One is you have to have a non-antibiotic selection marker. It has a 70 base pair antisense RNA. And then the other is it's designed to be compatible with that HyperGROT process, so you can make grams per liter of it. And then on the left is the actual effect of having less than 500 base pair backbone, and that is a combination of RNA-OUT with an R6K replication origin. And it's just showing that you've managed to marry the performance and production attributes of both the minicircle and the plasmid platform into one. Okay, so the top part of this is just sort of a cartoon showing how you can get away with not having to do recombination like you do with a minicircle. It looks complicated, but it actually is very simple from a manufacturing point of view. So on the top part is selection. And you see there's a constitutively expressed messenger RNA that makes a Levan sucrase, which is sitting in the periplasm. So you make your competent cells like you would normally. Now you put the plasmid in and makes that 70 base pair RNA, and it anneals over the start codon, the translation, and stops translation of the Levan sucrase. Levan sucrase is just a gram-positive enzyme that breaks down sucrose. And in E. coli, that, in the presence of sucrose, is toxic. So it's just very, very simple. You just transform and then plate them on sucrose plates, and the cells that aren't transformed die. The bottom half is how the replication works, and this has been wired to make it compatible with the HyperGROT process. So you can still do the reduced growth rate at 30 degrees, followed by a temperature upshift. And you can see that the rep protein is wired to be heat inducible. The reason we like the reduced growth rate at 30 followed by temperature upshifts is it's very generic. Many plasmids have instability issues, like an AAV. All those of you who work with AAV and you know about these ITRs and you get microdeletions and stuff, or lentize, that low temperature growth with a low copy number through the growth phase allows you to make those things stably. So that's what's really important. We want to be able to make all the classes of vectors with this. And this just shows that you get high yield manufacturing, in this case 2.4 grams per liter of a nanoplasmid. All right, and this is just very briefly showing that those minicircle advantages are captured. This is work that we've done. I'll show you later what this vector does in vivo. This is work we've done with the PVAX1 vector, which is really used heavily in the DNA vaccination field. And that vector has about a 2 kb backbone, and we made series of deletions. One, some people come to us and just have us remove the antibiotic resistance marker, because especially in Europe, people are quite concerned about having antibiotic resistance markers, so some of them have us do that. We've also done the full Nanoplasmid retrofit, and this is just showing in vitro results, and it's actually even more dramatic in vivo, that you do really improve expression in every cell line we've ever tested by removing that backbone. Now this is work that we've done with Dr. Harbaugh, Richard Harbaugh's lab at DKFZ, where Matthias did a transfection work where he was looking at plasmids versus nanoplasmids for activation of various innate response, I guess, as a generalization. And you can see a plasmid in this nucleofection experiment activated a lot of genes, but the nanoplasmid activated only a small number of them. And when you looked at minicircles, the nanoplasmid and the minicircle are about equivalent in this experiment. So again, you're capturing the advantage of the minicircle. My theory on this, I don't have any data, is that sort of consistent with all the work that Marquet's group did, that backbone is getting heterochromatinized, which is preventing expression, but also is causing a danger signal when it gets into the cell. So what you're looking at is a response to heterochromatinized backbone. That's a theory. I'm not sure that that's the case, but it would account for why we see this silencing in vivo and this kind of reduced perturbation in a transfection. So basically, the nanoplasmid it captures all those advantages of minicircles without the production issues. And so we do a lot of vector retrofits. And I'm going to just talk a little bit about what you can expect if you're like using like a transposon vector, as I say, continuing with the indirect vector theme. So we're going to solve these issues with this. And so this is an example of a transposon retrofit. As I mentioned at the outset, one of the groups that showed that you could get 6-fold higher CAR T cell production due to improved transposition and reduced toxicity was the Hudasek group. And this is just a slide showing when they compared nanoplasmids and minicircles, they saw equivalence of nanoplasmids and minicircles. So this is kind of a summary of what we've seen with different groups. The AAV one, that big improvement with AAV is with the SC AAV. I just want to emphasize that. So those have defective ITR termini, and probably reading into the backbone really, really damages the titers and production. You see equivalence to slightly higher with the minicircle and the nanoplasm with your standard SS AAV vectors. But of course, you get rid of any antibiotic packaging into viruses that you send into the humans. Lentiviruses, we've heard from people that are using our vectors, our retrofits, that you do get quite a bit higher viral titers. And again, I would say that's probably because you put it in there, you're trying to make virus, and you're reducing the toxicity and reducing the silencing, which allows you to make more viruses over more time. It's probably what happened. And we also, I'm not going to get into it, we also have made messenger RNA vectors. In those cases, of course, you're just going to linearize them, but our platform allows us to make a much higher, much better vector for producing poly-A vectors. Okay, so I'm going to end talking about where the plasmid is the final product, so the direct gene transfer vectors. And again, I'm talking about the PVAX1 vector, and sort of like I did with the indirect vectors, we're looking at where these backbones came from. So this is actually good. These are from the 90s, so they're only 20 years old. And one of the problems, though, they're all designed in the 90s, and about the same year the PVAX1 came out is when all the RNA-OUT work came up. So these were sort of created before people realized all the potential bad effects of transcription of the backbone. And so people have since then found that, in fact, the replication origin, the bacterial region, is extensively transcribed after transfection. And in the case of the PVAX1 vector, that eukaryotic promoter that is in the puck origin is actually oriented counterclockwise, so it should be making double-stranded RNA. And that might, I'm not sure about that, but it might explain one of the reasons why PVAX1 is a good vaccination vector. Its expression in vivo is terrible, but it's a pretty good for getting antigenicity. So it may be that it's got a pretty good adjuvant effect. Now, at the end, if I have time, I'll mention other ways you can get adjuvant effects without having to knock your expression down so dramatically. And this one I'm going to tie in with minicircles. You'll see this is work we did with Marquet's group and others as well, where we looked at nanoplasms versus minicircles. And I put up on the left, my pointer isn't working, but you'll see in the middle there a mini intronic plasmid. That the K-Lab came up with an alternate approach to getting the nanoplasmid-like effect. What they did was they put RNA-OUT and a replication origin into an intron, and then they had no backbone. And those in nanoplasm perform very similar. So we're not the only ones that have sort of seen a way to do this. They also have, using RNA-OUT vectors as well, a way to have high yield. And mini intron plasmids and nanoplasmids are very similar in terms of their effect expression. But if you look at the right, you'll see that the minicircles is quite a bit lower expression than the nanoplasmid. We see this a lot, and it really does correlate with the source of the minicircles. Minicircles are terribly hard to make, and as I said before, you got to be careful about your production process because of the purity. I think a lot of the results, because technically there's no reason why a minicircle shouldn't be as equivalent expression, but I think it's just that they're so hard to make that you never get a high purity product like you would with a nanoplasmid. And so one of the things we're interested in, because of the improvement with PVAC compared to PVAX1 and so many people using PVAX1, is this whole idea of using vectors for passive immunotherapy. And I'm sure many of you have heard about the whole concept that you currently inject antibodies which have a short half-life, and you have multiple injections of therapeutic monoclonals, and the idea would be to have actually a vector that you inject into you and allows you to make that antibody in vivo, so you have less injections, less complexity. And so in that thing, I'm not going to get into a lot of detail on this, is we've designed ways of making dual promoter vectors, so you can just, rather than trying to figure out how to do a 2A peptide and have it cleave properly, you can make it, this is just a couple of Avastin ones we made with both the CAG and the CMV promoter. We've designed ways to put together the two transcription units to ensure balanced expression of each transcript and stability in E. coli. Because if you just reproduce the transcription unit twice, if you just reproduce the transcription unit twice, you'll get instability, and it's very difficult to manufacture. So this is work that we did in collaboration with ICOR, showing that we've managed to, by putting together and making these the vectors, the dual vector thing with the differences that we've managed to manufacture, that we've gotten around the promoter interference problem, and you have good expression, in fact, equivalent or higher than a mix of the two individuals. That's really unusual with the dual promoter vectors. A lot of the dual promoter vectors are very hard to configure. Now, one of the things we developed, and I said I'd go briefly into into promoter things, is we've developed a muscle promoter. Virtually everyone working in the passive immunotherapy field is interested in the concept of having a muscle promoter, but virtually all the muscle promoters that are out there have less expression than constitutive promoters. And because you're trying to drive a level of expression of an antibody that gets you into a therapeutic window, no one wants to sacrifice expression. And so this is just showing, that's a CRM Desmond, which is the best I've seen out there prior, and then that we've managed to achieve expression. This is just one of our MCK Desmond promoters that's equivalent to the CMV in vivo. And this is work we did with KU Leuven, that basically we've tinkered with the vector, and we found that for muscle, that the CMV is good, the CAG promoter is better, but the muscle promoter, in this case in vitro into muscle cells, actually is predictive of in vivo. You actually are outperforming and getting good levels of expression with this particular promoter. Now, those are all electroporation experiments. So this is work we did within CellArt to use tetrafunctional block copolymers for delivery. And this is strictly their work on this slide, showing that they are getting very high levels of expression after delivery. And this is showing what happens when you use our muscle promoter vectors with their block copolymer delivery. And in this particular case, you have on the left, you have a reporter gene, luciferase. And you see in this case, you're getting about 3 logs improvement compared to PVAX1 with that delivery. On the center, you actually have an antigen, which is you're trying to make an immune response. So you look at it, you got 3 logs increased expression. You haven't done anything to the antibody humoral response. So this is important for this particular application, because that's one of the drawbacks of in vivo expression of a monoclonal antibody is the immune response. If you have the possibility of innate activation at the site and getting an immune response even to these humanized antibodies, in this case, we can really 3 log improvement of expression without changing the antibody response. So it would be promising to be able to use it for that application. Okay. So as I mentioned before, I thought maybe the reason PVAX1 works well for DNA vaccination because they're potentially double-stranded RNA production, which unfortunately really knocks down the expression of that. This is just showing that you can modify the vectors to increase the innate activation using RNA without having to knock down expression. The expression is not shown on these slides, but basically this is a modification. We have two different things that we put in. One is the RIG-I activating vector backbone, which is basically Pol-3 promoters making a double-stranded RNA, which activates the RIG-I receptor. And then on the vector map between the transgene and the poly-A, we've inserted a CpG hairpin, which generates CpG RNA. So they're very different. The RIG-I is a Pol-3 transcript with a 5' triphosphate, which allows it to activate RIG-I. The other one, it will be activating toll-like receptors through autophagy. And you see you get quite an improvement of T-cell response, and we've shown also antibody response when you put these kind of things into the backbone. Okay, so in summary, that's the nanoplasmid backbone. It was created to give a solution to the problems with large backbone vectors, both for indirect and direct clinical vectors. It's high yield manufacturing. If you've got a minicircle and you're happy with the result, you're going to get the same or better result with a nanoplasm because it has the same principle of mode of action. And I'll stop there.
Time for questions, and I just realized we're a full house in here, so you can yell out your question from your seat. Just want to climb over the person next to you. I have one, James. So one of the problems, I mean, we've worked with minicircles a little bit, and one of the problems we run into is the concatemerization when you do the induction, which may actually, when you're doing certain kinds of transfection, explain the stability of these things because you get these really large pieces of DNA. But when you're trying to do a nanoparticle, it's actually a very bad thing. You really do want a minicircle, not 20 minicircles stuck together. Is that a problem at all for the nano?
Well, one of the reasons we use R6K origin. If you look at the literature, it's when the R6 origin was part of the whole the PCOR system, which was used by Aventis in all sorts of clinical trials, and they actually put in the SIR region into it to get around the multimerization problems with it. How we handle it is this low metabolic process. So we keep the copy number low during growth. So we have what we call a copy cutter strand with more details, but we keep it very low during growth and then bring it up. And if you do it that way, it's fine. So that temperature-inducible R6K origin, if you just did a transformation at 37, you probably have so much vector in there that it would kill the cell, right? So yeah, you do have to use the lower temperature and maintain it at a low copy number. But if you do that, you can keep it pretty stable.
Can you speak up, please?
Sure. When you use polycationic polymers or whatnot with these smaller size vectors, say you're under like 1 kb, do you see that the nanoparticles end up much larger or smaller? Because right now, when you use smaller polycationic particles, you don't get under some like 200 nanomolar, nanometer. So do you actually see a smaller size nanoparticle with these when you work with anybody, or do these get as big like most of them?
We haven't.
Can you repeat the question?
The question is about polycationic nanoparticles. Unfortunately, I haven't worked with anyone on polycationic. Those are neutral block copolymers that we're working with in cell art.
Another question? So maybe.
Thanks, James.
Yeah, thank you.